We have two speakers tonight. Both of them are professors at Cal Poly Humboldt. I will introduce Dr. Lucy Kerhoulas first. She grew up in San Rafael. She received her BS in botany at Humboldt and also her MS in 2008. She received her MS working with Steve Sillett in redwood canopy physiology. And then she went on to get her PhD in forest ecophysiology from Northern Arizona University in Flagstaff. She's very glad to be back in Humboldt as an associate professor in forestry, where she's been teaching since 2013. And she says that her research interests include pretty much anything related to forests. So that is Lucy Kerhoulas. Our other speaker is Dr. Rosemary Sherriff, who grew up in Davis. She is a biogeographer with a BS in geography from the University of Oregon and a master's and PhD in geography from the University of Colorado in Boulder. She has taught at the University of Hawaii Hilo, also at the University of Kentucky, before she started teaching at Humboldt in 2009. She is a full professor and chair in the Geography, Environment, and Spatial Analysis Department. Her research interests include forest disturbance ecology, fire ecology, climate, and tree rings. So please join me in welcoming Lucy Kerhoulas and Rosemary Sherriff to our meeting tonight.
I want to thank you all for being here. We're really excited. Lucy, Rosemary, you don't have to remember that, but just for clarity. And Lucy and I have been working together for a number of years. I won't take a long time here, but we found that we really like working together. So we're going to tag team this talk in the way that we tag team research. So it might give you a sense of how we do things together. So we're going to focus on thinking about forest systems in Northern California. And one of the reasons why we're interested in this, because we all know that we have a very biodiverse forest ecosystem, among other organisms. And so we wanted to understand the context of the impacts of drought. All right. Uh-oh.
Let's see.
I missed.
Huh, weird. This is
how we do research right now.
This was working a minute ago. I don't know what happened. We'll just have to advance. Maybe we have to click on it.
Does that work?
Okay, now this is working.
Okay. I want to acknowledge that where this research that we're going to talk about, where we live and where we work at Cal Poly Humboldt, sits on unceded traditional, ancestral, and present-day homelands of many indigenous nations and people who continue to live and thrive in these places, such as the Wiyot, Hupa, Karuk, and so on. And so that's very important context for thinking about the lands that we're talking about.
Yeah, thank you. Thank you. Seems like if we make that go away, then it doesn't. Okay.
Okay. So what we thought was we would give you sort of some context of where we're going tonight in 3 acts. Act 1, project overview. Act 2 will be a study on tree drought response. And then the third is thinking about the demographic regeneration and mortality in these forest systems.
Okay, so or divide and conquer here. Okay, so I was going to introduce you to the setup for this multi-year project that we worked on. So we're condensing years of work into like the 30-ish or so minute talk. Okay, so why we focused on Northern California, this area is really complex geologically. So On the left map there, you can see the geology is complex. There's lots of colors. And even on the right, if you kind of zoom out and squint your eyes, and even trying to do that, it's still pretty complex. There's a lot going on in terms of the parent material in this area, and that's going to affect plants. Another cool thing about this area is that we have all these kind of nooks and crannies topographically. So we have these climatic gradients that are occurring at the regional scale. So generally speaking, as you go from north to south, things are colder and wetter up north, and then as you move south, they get warmer and drier. And then similarly, going west to east, if you are on the coast, it's cooler and wetter, and then if you move inland towards Redding, it's warmer and drier. So we have these kind of latitudinal and longitudinal climate gradients that are happening, and that gives some interesting kind of things for plants to work with in the area. And then you can layer on top of the complex geology and on top of the latitudinal and longitudinal climate gradients, you can also layer onto that these elevational climate gradients. So as you go from low elevations to high elevations, typically it will be warmer and drier at lower elevations, and then cooler and wetter at higher elevations. And so put all these things together, the crazy geology, the crazy climatic gradients, and voila, what do you have? You have the highest conifer diversity on Earth. So we in our backyard here have really crazy conifer diversity, world renowned, lots of different types of conifers. And being into trees, Rosemary and I thought it would be cool to take a peek at how some of these conifers in these diverse forests of Northern California, how they responded to the recent, it's not so recent anymore, but the 2012 to 2015 drought that was pretty epic and was really exhaustively studied in the Southern Sierra. But we were curious about, well, what happened in our neck of the woods? Like, how did the trees up here respond? We can't remember how we divided up the slide.
Well, no, I'll take it just because just a little caveat that I had this ambitious idea that we were going to study all of them or most of the conifers, and Lucy's like, no, we need to do this in 3 years.
I think on the original proposal there were 20. I was like, I think that might be a bit ambitious.
So we narrowed it down to 6 because that was the fundable amount that we could do. We got this funded. We'll talk about that a little bit. And then we had a time frame and people power, right? So we chose 2 coastal species, Sitka spruce and western hemlock. They have similar distributions in our area. And then we chose 4 montane conifers, sugar pine, western white pine, so 2 pines, and Shasta fir and Brewer spruce. So here in the map, you can see these are the coastal species, and then the montane. So just real quickly about it is that we were interested in drought, but we were also interested in how did these conifers respond in their habitat. So we sampled based on looking at each one of these species in a dry habitat, in what we'd call a moderate habitat, and the environmental context, and then a wet habitat of the same context. Okay, so in total we have 54 sites. So let's look a little bit about drought, which I know that you're, as Californians, familiar with this, but let's put a little context. This shows 3 different counties, so Siskiyou, Trinity, and then Humboldt, and the little bit about the variation in drought. All of them have seen drought, and so we were sampling during this time period where after the drought we could identify the influence of drought on these 6 conifers during the 2012 through 2016 drought. But in our Northern California area, the drought really was much more significant from 2013 through 2015, whereas it was longer in California in the southern distribution. So 2012 through 2016 in whole for California was the most severe in the last 1,200 years. And then, of course, we know that we've had drought since then that has also been very severe. This is during the time in which we were sampling, so we don't have a lot of data from this, but we are following up with some studies. So stay tuned, invite us in a few years again. Okay, so our main questions were for this study: how do conifers respond to drought? Basic question, but important. And how does the biodiversity of conifers? And then how did forest demographics, thinking about regeneration and mortality, respond to this drought? And again, it's the for Northern California, it's 2013 through 15. Yeah, were the main drought years.
Okay, so how did we go about doing, looking at, like, how do trees respond to drought? Like, how did we do that? One of the methods that we used was looking at tree rings. So coring the trees with a little kind of hand drill and pulling out a tree core that has this record of growth. So you can see kind of just some images here of our tree cores, generally about 5 millimeters in diameter, so not very big at all. It's not harming the tree. And then we can take those rings of growth and convert them into, like, picture a ring of growth around the tree. So not just one couple millimeters of growth, but rather looking at it as the donut of wood that was produced annually. So we can call that the basal area increment, or BAI. So that was kind of our metric that we used to measure growth. And then, so we looked at drought responses via 2 lenses. 1, growth using tree rings, and then 2, physiology. We were curious about, well, okay, maybe growth doesn't capture the whole story. Let's try to check out what happened physiologically to these trees. And so how we did that was using stable isotopes, and don't gloss over or panic, but so the stable isotopes, that weird symbol here, this is the stable isotope discrimination, and I'll explain that a little bit more in a minute. But basically you can grind up the tree rings into a powder and then analyze them and get an isotopic signature. And then from that isotopic signature, it's a little glimpse into how the tree was functioning physiologically during that year. So it's kind of a neat little trick when trying to figure out plants from years past. And then we calculated these metrics called resistance and resilience. And those terms, they sound really similar, and they're often kind of like used synonymously. But in here, we have really specific ways that we're talking about resistance and resilience. So resistance is just how the tree fared during the drought relative to how it was doing before the drought. And then resilience is how did the tree rebound? How did it rally after the drought? So resilience is looking at the post-drought growth or physiology relative to how growth or physiology were before the drought. Okay, so if stable isotopes make you nervous or uncomfortable, you can just gloss over and kind of like daydream about walking through meadows of flowers. But if you want to like take a little journey into the stable isotope world for a moment, in leaves of plants, there's this enzyme rubisco that does photosynthesis, and it takes CO2 molecules from the sky and fixes them into sugars for the plant. And rubisco is really choosy. It's really selective and kind of a little bit uptight. And so it really likes CO2 molecules with the light version of carbon, with a C12 atom of carbon. And that's the bulk of the CO2 in our universe. Like 99% of the carbon in our universe is C12. But then there's like 1% or maybe less of carbon out there that has an extra neutron in it. And so it's C13, its atomic mass. It's a tiny bit heavier. And rubisco doesn't like that. And so it will actively discriminate against C13. Except when the plant is water stressed, it will typically close its stomata, the little pores on the leaves, to conserve water. And that's great. Yay, happy dance. It's conserving water, but sad, no more CO2 coming into the leaf. And so when the stomata have closed under water stress, there's less CO2 coming into the leaf. And so beggars can't be choosers. Rubisco has to settle for more C13 CO2 molecules. And so we can use the amount of C13 in plant tissue as a proxy for drought stress, because beggars can't be choosers. So if the plant was drought stressed and it closed its stomata to save water, you'll measure then more C13 in the plant tissue. So with that, that's kind of how we went back in the tree rings and tried to reconstruct how stressed out the tree was water-wise during the drought. So here's just a cool little example of this, just to show you kind of, here was a normal precip year and then a drought year. And this is looking at some pines in the San Francisco peaks of Arizona. And on the vertical axis, we have the C13 signature. And so you can see in a normal year, the signature was down here, less C13. And then in the drought year, when the trees had to close their stomata a bunch, we can immediately measure that in the tree. Like, oh, look, the trees were stressed this year for water. So it's a pretty handy little tool in the plant physiology toolbox. This is kind of cool. Again, if isotopes make you uncomfortable, you can just zone out. But this is, I think, really neat. And a lot of people have shared with me that they found it was cool too. So I thought I'd just really briefly mention it to you. So we're all aware that CO2 in our atmosphere is increasing, right? And that's maybe contributing to climate change. So we can see that the CO2 is increasing across time. This is time on the x-axis here. So since the Industrial Revolution. But some folks are like, oh, well, you know, it could just be natural climatic variation. Like, who's to say that the increasing CO2 is actually from human activities? But if you recall that plant material has low C13 in it because Rubisco discriminates against it, and then we consider that fossil fuels are largely plant-based, by burning fossil fuels, we're burning a really light C13 signature. There's not a lot of C13. So across time, since the onset of the Industrial Revolution, we've actually, by burning fossil fuels, changed the carbon signature of our CO2. So as the CO2 concentration has gone up, we've seen this depletion of C13 in the atmosphere. And a lot of people think this is the smoking gun, pointing that the increasing CO2 concentrations are in fact driven by human activity. Anyway, take it or leave it, but I thought we'd take that slight little detour with carbon isotopes. If that didn't make any sense to you, no worries, we're moving swiftly along. And so another thing, just kind of thinking about how we're going to interpret our findings that we're going to share with you. I know you're all on the edge of your seats to find out what we found. But before we share those with you, just how do you think about, you know, one of the questions that we had was drought response. How did the conifers respond to drought? And then another of our questions for this study was, you know, what were the demographics in response to this drought? So bigger picture of NorCal, you know, what was going on after the drought with forest mortality and forest regen. And so to get at that, we have to kind of think about how do trees die with drought, right? With humans, it's easy to say if you're alive or dead, like, do you have a pulse? Do you have a heartbeat? Are you breathing? But with plants, it's a little more gray, right? And so we can better understand our data and the things we're going to share with you, the findings, if we kind of think about how they affect forest demographics. So there's 4 different ways that plants can typically die in response to drought. And I'm not going to really go into the 3rd or 4th one because it's not that relevant to our findings. But the first 2 are kind of a big deal. So the main 2 ways that trees can die in response to drought are going to be hydraulic failure, where all of the water columns in the tree snap because they got under too much tension under water stress, and then the tree is ineffective at slurping water up anymore. Its hydraulic capacity is reduced. And then the other one is carbon starvation. So imagine if those stomata close under water stress and no more CO2 is coming into the leaf. If that persists for a 5-year drought, that can lead to carbon starvation for the trees. So that's kind of the overarching architecture of how drought and forest demographics might relate. Do you want to explain kind of?
So all right. So when we talked about mortality at some level, and one of the things that we're looking at, again, demographics, thinking about regeneration, new trees, and then mortality. So we, 54 plots. And so what we did in terms of mortality, we're not going to spare you with the whole sampling scheme at the plot level. But what we did is, the number of trees of live, number of trees. So the percent per area, in this case, hectare. And then we looked at seedlings, so the littlest ones, and then saplings per hectare. And then we dated the mortality and regeneration. So we actually used tree rings to date the ages of the trees. And I just want to emphasize this, a lot of this work we'll share is graduate students doing this, and this is enormously labor intensive, but really useful information. And not that many people actually date the dates of trees. Okay, Act 2. Okay, we wanted to pause. That was a lot. We're really
Let people regroup.
Questions, thoughts? Paul?
The C13 reduction over time because of the increased plant generating carbon dioxide after, that gives you a moving target, right? So does that affect how you read the
Yeah,
secret
definitely. That's a very astute question. You could be an isotopologist.
After being a retired professor.
Yes, you do. And you that Seuss effect, or the depleting of the C13 signature in our atmosphere across time, has to go into the calculations when we do this isotope work in tree rings. Yeah, very, very good question.
You mentioned the problem of how you tell when the tree is dead.
Yeah. Oh, how do we? It's a little gray, you know. We have a rubric and criteria in our, so basically it's kind of like a $1 million question, and there's different approaches to it. I forget our exact criteria.
It's actually kind of a hot debate. It's a very hot debate. And I've seen pretty heated discussions in conferences of big audiences. Interesting enough. But though, for example, if we take the tree growth data, we core the tree, and we know the end date by cross-dating it, meaning, you know, cross-dated against other tree growths. And then we look at, are there any needles? What's the bark look like? And so on. We can calculate that, but we don't always know what the mechanism is, which was Lucy was emphasizing. There's these 4 main mechanisms. We know that the tree is dead during that certain time period, if that helps. It's a fuzzy world out there in the mortality industry of trees.
I think we had, like, in this field work was now years ago, so I'm like, I'd have to go back and reread our methods, but we had a really specific criteria that Sophia Lemo, and she actually got a scholarship from CMPS and came and gave a talk like not too long ago on some of her work, but she had some very specific criteria of, like, if it checks these boxes, consider the tree dead. But I don't remember the details of what all those were at this moment.
Okay, you guys ready for Act 2?
Yeah.
Drum roll.
General, tree guards. First, we want to acknowledge that, or is that for the
Yeah, we're gonna psych you out a little longer.
Oh. We had a huge team, and this is just the first year, and we had a number of researchers and a lot of graduate students, and Lucy's youngest child right there helping with field work. And so we just want to highlight a few things. We have had a lot of funding for this study, including the National Science Foundation, but also the Native Plant Society, and then many units, I would say, you know, organizational units at the Cal Poly Humboldt. And in particular, we want to highlight, just for time, I'm not going to highlight everyone, but I do want to highlight these 3 individuals, which were the graduate students whose work we're going to talk about specifically, and then a lot of other people who've helped and continue to be involved in the work. So we want to be really cognizant that it isn't just us, and it takes a whole rodeo to get this kind of work done. Okay. Here's some results, right? Drum roll. Okay, so let's look at this graph for a minute. What you're looking at is this measure of kind of a volumetric measure of growth on the vertical axis, basal area increment. So you know, smaller growth to larger growth, and then years. And we're just looking at a period of time in recent history, so 2002 to 18, which is the last year of growth that we had, because we started our sampling in 2019. And I wanted to mention that we carried out this study during the pandemic, and we were able to successfully complete it, although it was quite complicated. We were able to, and that's really the result of all of that team of people that I mentioned before, that we were successful. So that was really amazing. Despite the pandemic. And so the main things I want to point out about tree growth is that you can see that the 2 coastal species, and it's an average, the blue line is an average, and then these are min and max, and you don't need to worry about that. But you know, what we saw actually is coastal trees in general grow larger than the interior, more montane species. Maybe not surprising, right? We know we get a lot more wetness, we got a lot more moisture on the coast, but that is important. And then the second major thing I want to point out is this is the major drought period that we were looking at. If you recall that drought that you might have lived through, what we saw is really rapid response of the coastal trees of decrease in growth. So they were responding very rapidly and decreasing their growth in response to the dry conditions. In contrast, the 4 montane species just kind of plugged along, very little difference in growth. And then the second thing that's interesting is that after the drought, we see this real bounce back. We don't have a lot of years because of the sampling time period, but bounce back. So really rapid response, both decreasing during the drought for coastal species and then a rapid increase in growth back. Whereas those montane species kind of like just continued to plug along. And we'll reflect back on that a little bit later as what that might mean in terms of potential for mortality of those 2 different types of mortality mechanisms that Lucy mentioned earlier.
Okay, so this is the growth side of the story, but sometimes the growth side of the story doesn't tell the whole story. So it's important to also, maybe if you can, take a peek at the physiology. So that's what we did next. Our 2 lenses of how did trees respond to drought. We had growth, and then we took it the extra mile and looked at physiology. And so we see a really similar trend here. So on the vertical axis here, we've got that weird C13 situation. So up here is less water stress, and down here is more water stress in terms of what the isotopic signature means. And so we see montane species kind of chugging along here, not too affected. Like they didn't really change their march. They didn't really change their beat during the drought. They're just kind of like whatevs as far as their physiology went. But we see, just like we saw in the growth for the coastal species, this really pretty pronounced change in their physiology. So we can say that the coastal species are much more sensitive to drought compared to the montane species, which seem to be just kind of rock steady. Like they just don't really change their tune. They do their thing. Whereas the coastal species seem to be more plastic in terms of their physiology and then also their growth rates. And so what can be happening here, what we can think about as far as kind of Rosemary was alluding to and how this dovetails in with our next set of questions about forest demographics and mortality specifically, is what does this mean like for how, if we think of all the global climate predictions for the future, things are getting warmer and drier. Like, so this, drought is here to stay kind of thing. It's the new norm that plants will be dealing with. So given these very different responses that we picked up, both based on growth and physiology between coastal conifers and montane conifers, like what does that maybe give us a hint into about how forests might fare across droughts in the future? And so what's happening here, the reason that we could measure this change in C13 signature for the coastal species is that when things got dry, the coastal species were like, and closed their stomata. And so we see that there was more discrimination against C13 throughout the drought. And so this means that if we put on our thinking caps about plant physiology, that if there's repeated long droughts, this type of a physiological response to drought could make coastal species much more vulnerable to that mortality mechanism of carbon starvation, right? They're starving rubisco for CO2, basically by closing their stomata to save water. Whereas on the flip, the montane species, by not really changing their stomatal regulation strategy in response to these more arid drought conditions, that puts them at really big risk of blowing their vascular system, basically of hydraulic failure, because under a lot of water stress, if they don't button down the hatches and close those stomata, that can lead to like runaway embolism in the vascular system, and the trees don't function hydraulically. So we have these 2 different responses that could lead to 2 different mortality mechanisms in these different groups of conifers, which is kind of interesting if you're into geeking out on trees. And then we thought we were joking, we're worried a little bit that this talk might be like death by graph. so we had to be very, but for us, we're like, oh, we definitely need to put that one in, so we've saved it to just 2 of these interaction graphs that we thought were so interesting. We had to share with you. So what we have going on here, and I know this Zoom bar is like hiding it, but basically here we have water year, so dry climate and wet climate. And this is for coastal conifers. And then here we have the C13 signature. So high water stress down here, low water stress up here. And this slide is looking at how did stand density play into the conversation, right? Like we have like really dense stands, especially on the coast. Picture if you're familiar with like Redwoods Rising or something, you know, Toothpick National Park instead of Redwood National Park. We have these dog hair thickets of really dense forests. Like how does that play into the mix of these drought responses? So here we're looking at density, forest density, and how that played into drought response. And so what we see here is that the high density forests in green, they had more water stress under dry years, under dry conditions, whereas under dry years, low density forests had way less water stress. So this is indicating that, hey, heads up, folks, in coastal forests, really dense stands, that contributes to increased drought stress in trees. So it's good ammo for like, yeah, we maybe want to go in there and do some restoration thinning in these dense coastal forests because that might alleviate some drought stress that we measured through this work. And then the second interaction that we thought you just couldn't live without was kind of the same sort of setup as far as the axes go. So we have water year down here with wet and dry, and then water stress as measured by the C13 signature, so low water stress, high water stress. And instead of density here, and this is for montane conifers, instead of density, the different colored bands here represent tree size. So if you think about, well, how we just did the previous slide, we're wondering, how does stand density affect these responses to drought? Well, similarly, let's talk about tree size. Like, how do large trees and small trees respond to drought the same way? Because on the one hand, you might think, hey, large trees, they're way bigger, they have way more internal storage capacity for water. Does that give them like, does that buy them a buffer? You know, and they also probably have much deeper root systems that can tap more water sources than say really small trees that don't have a lot of internal water storage capacity and maybe have some kind of underdeveloped root systems. But on the other hand, large trees also have these big crowns, and the crowns of the trees are the biggest water costs. They're really expensive when it comes to water because leaves are the #1 place that water is lost from the plant body. And so being a large tree means that you have higher water demand. So is that a bummer when drought rolls around? Well, for these montane conifers, yeah. It turned out that the large trees were much more sensitive to dry conditions compared to the small trees. So this is really good information to have. Like in a lot of the Sierra studies, it was the large sugar pines that were dying in response to the drought. So showing that in some of these California montane systems, the large trees are more vulnerable to those dry conditions compared to the smaller trees. Okay.
Okay, so just to hit some of the highlights for the drought study that we did on conifers, both 2 coastal species and 4 montane species. This is for coastal species. What we found is that coastal species were more responsive to drought. So resistance again means during the drought. So we found that there was lower drought resistance. They were much more responsive in terms of decreasing growth and changing their physiological conditions, especially, again, Lucy just highlighted that that's true in dense stands. That does support forest management actions to thin out forests that, not old growth forests maybe, but like second growth forests that have been impacted by prior land use changes. Second, but we saw this bounce back after the drought. So that's really interesting. And we'd like to follow that further as to what happens afterwards. The trees respond relatively quickly. This means, as Lucy pointed out, that they might be vulnerable because they're responding so rapidly to more prolonged recurring droughts. That would be carbon starvation for coastal species. In terms of the 4 montane species, what we find, remember, if you recall the growth or physiological graphs, they just kind of kept on plugging along. So they have higher drought resistance. They're not responding in terms of growth or physiology. But that means that over time, if you have longer droughts, they might be, and more severe droughts, they might be more vulnerable because they're keeping their stomata open and more vulnerable to water loss. We also, interestingly, we're going to show you, we spared you another graph, but what we found is that growth and both physiology of the montane species were negatively related to competition. So the more competition around an individual tree, the lower the growth overall and the lower the physiological carbon 13. Great. It's a regroup.
Any questions?
So you're not studying the low elevation inland species. Those are the ones that I would have thought would be the kind of the most subject to Drought.
Some of the really low elevation conifers, sumptuary,
Yeah,
yeah.
yeah, that are not a coastal.
Yeah, they're not coastal. Well, again, it goes back to Lucy wouldn't let me do alternate species. Yeah, but no, that's a good point. We had to selectively decide. And so what we chose were species that we knew were vulnerable based on Southern California and what's happening in Northern California where we have more biodiverse forests. And we'll show a slide in a minute where it shows like we have had lower mortality because we have more biodiversity in the Northern California. So you're right that we don't have those low elevation species, and that sets us up for studying those trees
And
too.
you also not have low elevation trees of the species that you have?
We have variation in the elevation because we have a lot of sites. So but we only, again, time, money, and hands on deck. We have 54 sites and 9 sites per species. So we have a limited amount, but we did try to vary the amount of elevation, and that relates to that dry, moderate, or wet habitat type in some cases. So yeah, lots of good things, lots of good variables that are like in our study or maybe should have been, right? Yeah, a couple of hands here, yeah.
Is that value, value that mentioned delta carbon 13, is that a negative value?
Yeah, the scale is weird. It's a negative scale.
Thank you.
Yeah, it's a negative scale. It gets a little weird.
Thank you.
Yeah, I wasn't gonna go into the delta notation. But yes.
Did you happen to find the correlation between size and physiological response for the coastal species?
We didn't.
We were just looking at density. Yeah, I was curious of maybe like as far as management recommendations, if it makes sense to target maybe against our judgment larger trees or smaller trees or.
Yeah, I think that's a product of our sampling. Like our methods were for this, that we sampled dominant and co-dominant trees. So in the coastal forest, to find like a dominant or co-dominant western hemlock, it had to be a big tree. So I don't feel like we had layers in the montane trees. We had a much bigger spread of sizes. I feel like in the coastal species, at the end of the day, our sample ended up being like really predominantly large trees. So statistically, we couldn't really pick up a size related trend because they were like all big. Does that make sense?
Yeah, and when we looked at the size, just to reiterate, when we did end up looking at the size distribution for the coastal species is much narrow. They're similar compared to the montane species per species. Yeah, so I said we didn't, but it's true that we probably just didn't hit the broader range to find it. That is important. But again, I would suggest that, and again, this is just like conjecture, that it's much wetter on the coast, and so we may not find size as much of a difference. Yeah.
I guess related to that, when you're in your plots, you talked about density, but you didn't really talk about size distribution within the plots. Talk a little bit about that.
Yeah, so we skipped over a lot of detail, and one of the things with this study is that we sampled, rather than like normal plot where you're sampling all the trees in a plot, we called it a plot, but really what we did is we centered that and we focused on trees. So our sample unit is really the tree and then what's around that tree. And so it's not actually like, carrying a distance tape out and then sampling everything. It's like a distance around each individual tree as our sample focal unit, if that makes sense. So the focal unit at each plot is just a tree, and then what's the competition? We calculated the number of trees and the density around and the diameters of those trees around that individual tree, so that we could then sort of like scale up a little bit. But then to go further a little bit, which is that because of our limited people power and so on, we chose only the mature trees. We didn't sample all sorts of different sizes of trees, and we recognize that that is something that we need to make sure that we're pointing out, is these are mature trees. They're not all of the size of trees. So thank you.
Rosemary's thinking we need to skip some graphs. So how you guys feeling about graphs? You can be honest. We can handle it. Okay. Okay, we'll move swiftly along here. So Act 3, demographic trends. Okay, so this is our, we had the two questions. One was drought responses, and we used those two different lenses, growth and physiology, to try to better understand how conifers in our hood responded to the recent drought. And then our second question is, well, okay, let's take a step back and just look more broadly at, you know, demographics, like trees dying, trees regenerating, what was going on.
Okay, just a little geographic context on the geographers, then we can take this slide. So let me use this for a second. So you can't see the years, but basically this is a graph showing number of dead trees that have been modeled in California from 2010 to 2018. So 2010 to 2014, what we see, the drought started in 2012. What the data show that there's a lagged effect of mortality. We saw a lot of mortality in California, but it was like 1, 2, 3 years after the drought started. So we see this peak in 2016 across California, which is interesting, right? And so, and our sampling period is during this time period, right? So, but one of the other important things to look at is the colors in this map of California. And so the colors represent the forested areas, and the red and orange colors, the darker colors, represent more mortality. So we see a lot of mortality. When we heard all of the publications of in the news of the massive mortality, really was concentrated in the Southern Sierras. We see pockets of mortality in Northern California too, but it was a lot lighter. And in fact, we saw more mortality during 2020 to 2022, in fact, than during this time period. So why? Well, our biodiversity, right? But that sort of set up why we wanted to look, like, well, what's happening in Northern California? Which trees are dying and what's going on in that case? So much more diverse conifer response in Northern California. And just a couple of trends here in terms of mortality. This map shows, you can't see the legend based on the thing that's blocking it, but the larger the circle, the darker the circle means higher mortality, okay? And the red line represents the distribution montane on the right side of, those are our sample sites in Northern California. And then these are our sample sites in the coastal species. So note the big circles down in the southern distribution. That's the southern edge of both Western Western Hemlock, thank you. It's a little late. So what we see is, right at the border, the southern border, the driest areas, right, may be expected, but that's where we found the most mortality. Another geographic trend, I would say, but we don't have a lot of data to support this, is that if you look at the eastern side, also dry in the montane species, we see the biggest circles here. So maybe suggestive of, again, like an eastern dry sort of edge range distribution. We also, this isn't showing it, but the pines, the montane pines, are the ones where we see the most mortality. This is where I was gonna press forward. I'm gonna say this quickly for those of you who like graphs. This is the statistical analysis that looks at real simple, what's the relationship to the mortality date of death, okay, and dry or wet years. So on the vertical axis, more dry, less dry years. And what we found, the black bars represent, and you can't see, oh yeah, you can see it. This is the death date year, 0. What we found is statistically significant, yes, dry years are associated with mortality dates. And that we see this relationship kind of after that's probably an autocorrelation, thinking about there's many years of drought, happening. And so this supports the graph that I showed you, the geographic distribution, but it really points at site level, and we have species specific level of, the drought contributed to prior drought, prior drought contributed to the death dates here. Okay.
Okay, so regeneration, so forest demographics, we have like two sides of that coin. We have the mortality, and then we've got the regeneration, which hopefully is like a tale of hope. But here we have kind of how much regeneration was detected across our 54 sampling locations, and these are bins of decades. And so what you can see here, this is the drought period, the 2012 through 2015. You can see there was really little regeneration detected across our 54 sampling locations. And then during the drought, post-drought right here, which I wish I could see that year, but I think it was like 2016 through 2020. There's like no regen that we detected. So when we went and sampled the seedlings and saplings across these 54 locations, and then cut them off and, under a dissecting scope, counted the rings and dated them to what year that small tree had regenerated, there were like none that were regenerating to here. So a little concerning given the mortality that we were measuring, and then not measuring a corresponding, like you think, oh, when trees have died, that has opened up some real estate, and there's going to be new trees coming in. But given the drought and the post-drought conditions, we didn't really, sad to report, measure a whole lot of regen, which was a bit concerning. And then similar to the analysis that Rosemary showed really quickly about like how there was that lag with, up to 3 years before the death year, dry conditions, similar story with regen when we did a similar statistical analysis. So what this is showing, here's lagged from tree establishment year. So after we dated all of the tree establishments, then we layer on climate data. So here 0, right, is the year the tree established. And what this analysis showed was that the year prior to regen was significantly wet. When we looked across all of our regen data and layered it with climate, time and time again, statistically, the year before successful establishment was significantly wet climatically. So just saying like, oh, maybe we're not measuring regen, during the drought or after the drought because we need these wet conditions to have successful germination and establishment of new cohorts of trees. And this was for the montane, these data. Okay, so did you want to do the summary? You're a little better at them than me.
That's true. Okay, summary. So if you're like graph, like, whoa, okay, this is what we really said, is that the tree mortality data that we found, both, in this case, we did the plot level tally mortality, and then date that mortality. We found the highest mortality in the pines, sugar pine, western white pine, and the highest mortality during and after drought, right? To that after drought too. Regeneration, the seedlings and saplings, the lowest within pines. We didn't show you a graph of that, but that's really what the lowest starting with the graph. And the highest regeneration, the most regeneration were in the shade tolerant species. We know that we, due to lack, fire suppression and so on, we have denser forests in many places for a variety of reasons. And we saw high regeneration of shade tolerant species. Those were fir spruce in our study and true firs. And then regeneration during and after drought was notably low. And when we did find regeneration, those dates occurred after wet periods. Our take-home points. Let me take that.
Okay, I'll take it home here. This is like a summary of everything that we've been talking about for the past hour. So if you've glossed over or tuned in and out of isotope dreamland, here's kind of what we've said, distilled down to 4 bullet points. In our regional conifers in Northern California, we found, like, good news. When you take a few steps back, I think our data show that the conifers up here, compared to say the Southern Sierras, they seem pretty darn resistant and resilient to the recent drought. We'll see how things unfold moving forward, but this one case study that we did, all things considered, the conifers showed pretty high resistance and high resilience. The coastal species were more sensitive to drought than the montane conifers. They were more sensitive. They were more responsive to the drought compared to the montane conifers, which are kind of like slow and steady, just doing their thing, and they don't seem to really deviate from that beat. We found overall, again, kind of taking a few steps back from the nitty-gritty, lost in the weeds of our particular study, looking at, compared to say the Southern Sierras or something like that, mortality was pretty low in our regional forests in response to this 2012 to 2015 drought. So on the whole, it was like less than 20% of the trees. So it's not too shabby. Like our NorCal forest did pretty well in that drought. Somewhat like alarmingly, regeneration since 2013 has been very low. And the things that are regenerating are mostly shade tolerant species. So this could really be the harbinger of like a big looming on the horizon shift in species composition for a lot of our NorCal forests, from a lot of the pines to more shade tolerant firs and spruce. All righty, I think we've done it. So you can ask us questions, yeah.
So, question, were there something you learned when you got into all the analysis that was surprising to you? Was it surprising to you that the pines were harder than other trees?
Yeah, I'll just speak real quick and let Lucy. I think one of the debates that's out there is, are wet sites gonna be, or wet, species that are adapted to more wet environments gonna be more sensitive to drought, or dry, that are already in dry environments? So one thing that was interesting in terms of growth and physiology is we found those coastal species to be much more responsive, and in particular, the southern distribution, thinking about that. And then I would say second, we found that the montane species resistance, like the fact that they weren't really responding, was interesting, because the lack of trend was interesting. And so what does that say? Are they going to be fine? Are they going to be, what does that say for the future? I think that I'm what you should say.
Yeah, I was surprised at how rock steady the montane conifers were, and like they were unfazed. Like there wasn't really a measurable change in their cadence, whether we looked at the growth lens or the physiology lens, which was like, oh, that's weird. But I think then we did see pretty high mortality in a lot of the pines, which were all montane. So maybe that strategy wasn't so effective in the long term, because we saw really high rates of mortality in a lot of the pines, which were in that group of montane conifers that seemed to not really change their beat in response to drought. So kind of suggestive of possibly a lot of hydraulic failure leading to mortality in those species. And when we were putting together the study, like thinking, trying to leave no stone unturned and like think about all the different things that we should have on our radar, like Rosemary was saying, we were like, I wonder how wet, like trees in wet portions of their range, say we just focus on like one species, say like, sugar pine or something, sugar pine in wet locations versus dry locations, like how will they vary? Because on the one hand, you can think of trees that are in wet areas, they've had the good life, right? They are like really cavalier with their water use, even at the leaf level physiology. They're just like, whatevs, leave their stomata open. They don't have high water use efficiency. And so then if things get dry, a lot of times they're the ones that are really heavily hit, even though they're in this like dreamy wet environment. Whereas the plants that have been growing in dry conditions, a lot of times have like acclimated physiologically to be really miserly with their water use. So they're a lot tighter with their water use efficiency. And so they often can do better, even though they're in these kind of drier, harsher conditions. So it was kind of interesting to see, because we weren't sure how it would play out, that the coastal species that are in these dreamy, wet environments, that they were so sensitive to the drought. That was kind of interesting.
Oh yeah, I think you're right. Yeah.
So our northern area had a low mortality rate of less than 20%. What was that number for a high rate, the highest spot?
Of our study?
No,
Or
just
the-
like in
Oh yeah,
lower Sierra.
there's some high 90s.
I think there were some areas where it was like 100%.
Yeah, areas are more homogeneous in terms of species composition than what we have up here. As you've traveled around, we have much higher diversity overall, species composition, whereas in Southern California, generalizing you guys are all but we get more homogeneous stands of species and or pines, right? So
Also up here, where kind of, if you remember back in our tour of tree physiology, that there were these 4 potential mechanisms of like the coup de grace for tree death. And I didn't really go into the 3rd and 4th ones, but in the Southern Sierra, those biotic agents played a huge role in mortality. Whereas up here, we didn't really see that so much. But in the Southern Sierra, where it's really pine dominant, there were a lot of pines, the bark beetles came in. It's when the tree is drought stressed, It doesn't have as much photosynthate for making resins and defensive compounds and stuff. And so it's really, it's like a sitting duck for those bark beetles and different pathogens to come in. And so I think that contributed a lot. So down in the Southern Sierras, there were certain small areas where I think it was almost like 100% mortality, especially if it was all one species and the bark beetles just came in and like decimated the situation.
Just a couple of nuances, and I'll stop talking, that, it does relate to species composition. So, you know, if you have a more homogeneous, you're gonna get a bigger outbreak. You tend to get the smallest trees, like the advanced regeneration, trees that are under the canopy, of the same species that survive, because the beetles need the larger trees for their food.
One thing just to tag on that, just to say, Donna, in response to that, what was surprising from Sophia's work looking at regeneration, I was blown away. She was collecting these like tiny little trees that were like this big, and under the microscope, looking at them, they'd be like 75 years old. I mean, so just it gave me an appreciation for like, whoa, these things have been in there biding their time. Yeah, so that was really eye-opening. I thought like, oh, maybe 10 years old. It was like under the scope.
Okay, fine.
I feel like I might have missed this slide. Did the drought conditions express itself similarly at elevation as at the lowlands?
Yeah, great question.
In terms of precipitation.
Yeah, you didn't miss, well, we didn't put in that, but in the criteria of the sites, I won't go into too much detail, but we have, but what we did is we made sure that the drought was droughty in each one of those sites. So it was lower than a certain value, meaning that it met a drought criteria.
And approximately what is the?
Yeah, so we use the Palmer Drought Severity Index, if anyone's familiar with that. Yeah, okay. Well, the graph that I showed of the drought, the value, the orange, the darker orange to lighter yellow, or nothing, it would have been the darker orange.
Like,
So,
what were the stress indicators or the drought stress indicators that you would use to sort of stage the drought level?
oh, yeah, so we use severe or very extreme drought as the period of time that we know in the counties. And then at the individual sites, every site that we chose had a value that meant that that site, regardless of if it's a wet habitat or a dry habitat, met the drought criteria. And what I mean by that is like, it's a measure from looking at the elevation and the climate data combined, and looking at that those years each were dry, and we did an average of the years from 2013 to 2015. Meteorologically, yes, but
Not necessarily like hydrological.
Well, we used the Palmer Drought Severity Index,
it's a
PDSI,
good question.
which is kind of like this magical silver bullet of like a drought index. To me, it's a bit of a black box sometimes. Like
Well, there's different measures we could have used.
Yeah.
We looked at, you know, the 30-year average, and then what was the year, but we ended up using the Palmer Drought Severity Index. So we could get into the full discussion of which one's better and so on, but that is a standard, and so we felt consistent, and it was used, it's used in other studies.
Okay, just for the sake of the audience here, the reason why I'm asking this is you don't see a drought response at elevation. Maybe the conditions were different up there, so that's why I'm asking.
No, it's a really good question. I think if you want, I think we have some more graphs hidden at the back that I could scroll through, but that has like some PDSI values to show. But we were
Well, what I'm saying that you're right, that at higher elevation, you would expect in some, unless it's really dry, like a south facing aspect, that it, you know, a north facing aspect might be less dry. But we checked that. So even though, that's what I mean, like we checked that. So whatever the habitat condition is, that site was dry for that site compared to the, you know, 30-year average. And then also for this drought index that is commonly used, but really great question.
Yeah. So
Because there are many different metrics we could use and so I appreciate
one of
that.
our criteria in selecting for each of those 54 sites was, did it get droughty? So like we specifically checked that to confirm.
And some
like,
sites
yep.
didn't, and just to go further, like there were many sites that didn't meet that criteria. And we were like, okay, that's interesting. A lot of our sites, we didn't have as much mortality in Northern California. We didn't meet that as much. And maybe that, that's the reason, but these sites that we looked at did meet, this particular criteria. Great question.
Where you had your really poor regeneration during the drought, did you also have a lot of competition though at the same time from shrubs, or were the shrubs also kind of kept low? Because I'm seeing right now, of course, really bad competition from shrubs and burns.
Yeah.
Mhm.
Then I was like, how are those trees ever gonna make it? There's only so much water, right? That's available. And the shrubs are taking the sun and they're taking the water.
It's a really good question, and from off the cuff, from what I remember, was that the shrubs were much more of an issue in the coastal sites, weirdly.
Yeah.
Like the huckleberries, I remember the crew just being like, the huckleberries, like in the salal, just battling it, where it's like over their heads. So I feel like the coastal shrub situation was more, and part of our study, which again, we had to, so many things to share with you, so little time. We didn't share with you all of our selection criteria, but one of the things that we didn't include in possible sites for this study were any areas that had burned within the last 50 years, because we were just like, we can't layer on fire, like we're already kind of doing a lot. So we are, stand by.
We are.
Yeah.
Fire and drought, but I'd have to look, and Sophia's thesis is published from the Cal Poly Humboldt. You can get online, but we also are working it towards a published manuscript. I don't recall that shrubs, because we did take that didn't come out as a variable in the statistical models. Yeah, but competition, density did, basal area or whatever, some measure of the plot scale did, of density,
For the trees.
for the regeneration too.
For the regen.
Yeah.
But tree species.
Basal area, like density of trees.
Density of trees.
Overstory
Overall
trees,
density.
yeah.
Yeah, but not shrubs.
But not shrubs.
I mean, we have a sample and maybe that shrubs are very important. We know shrubs are important, but we didn't find it.
And maybe if we had burned sites, shrubs would have been like more of a force to be reckoned with, but it didn't come up like to the top of any of the models or anything. Good question.
I was wondering what species you might look at next, and whether you thought of maybe ponderosa pine and Jeffrey pine.
We are looking at ponderosa pine. What else are we looking at?
Okay. So many conifers, so little time.
What? That input.
Okay, so right now our study has, we have
Jeffrey pine, Jeffrey pine, ponderosa pine,
sugar pine,
is one that we see a lot more time.
Doug fir, incense cedar. There's one more. Forgetting now.
Western, I think we didn't do Western white. I mean, we had to select.
Yeah, so many conifers. Yeah, I can't, it's like terrible, but we're only in the first year of it.
Yeah, we're examining.
We have one more species.
Yeah.
What's that? No, it wasn't red fir.
Part of the problem is, you guys, if we get real nuanced, is that we're looking at a fire footprint, right, to keep it consistent. So we're looking at one, we've selected 2008 because it was a big fire year. And then we've got the drought, after that. And so what's in that fire footprint is that, where we could find like severe enough fire and then no fire to do a decent study comparison. So we've searched for a variety of species. We're justifying it now that we didn't sample all the species, but yeah.
Yeah, it's kind of cool. So we're looking at a proof of concept for pyrosilviculture. So using the 2008 fire, the hypothesis being that exposure to wildfire can be a management tool that will increase drought resistance and resilience. So using stands that burned and didn't burn in the 2008 fire season, we then can go back and look at via tree rings 2 droughts, the 2013 to 2015 drought that we've just been chatting about, and then also the 2020 to 2022 drought. So we have successive droughts, and the hypothesis would be that trees that were exposed to the wildfire have greater drought resistance and resilience to those 2 droughts compared to trees that didn't have wildfire, because the fire is moving in and in theory reducing competition. for those trees. So to be determined, you'll have to come to another CMPS talk. And hear about it. Okay.
Well, that's.
It's a few years out.
Two questions, I'm hoping they're not too, long, but, the first question being, in terms of the rebound and needing wet conditions, were those conditions like significantly wet, compared to historic, precipitation, or just significantly wet in terms of like, compared to the conditions that have been in?
Do you mean for the regen, where we saw the year
Yeah.
before regen? Yeah. Okay.
Yeah, what is really
Do you wanna
wet,
explain
like
that?
historically really wet, or just really wet for that drought period?
Yeah, great question. So let me answer that first. So with that kind of approach, you need to look at the conditions. If you were looking at the 1980s to present, it's out of context with the regeneration period that we're looking at. So we use the regeneration period, but that is actually quite long. So I'm just going to use that as an example. Regeneration is some of those trees went back like 80 years. So we did actually use the historic period, but we have to bound it within the time period in which we're looking at the regeneration. Does that make sense? Like, you don't want to look at a time period that's not where your regeneration is. Basically, it compares real quick, sorry. The regeneration years of establishment compared to non-regeneration, as well as we do the simulation modeling. And then it compares and it shows that it's significantly wetter in the year prior during those regeneration years compared to the years where there was we didn't find regeneration. If that made sense.
Okay.
I'm
so it's
happy
not
to go
like
into
there
more detail.
The wettest conditions in the last 80 years.
No, we don't know, we didn't look at the level of wetness, it's more like, was it wetter than? Yeah, it's sort of that kind of comparison.
Okay, and then my other question was, do you have preliminary data for regeneration and or depth for 20 and 22 so far?
No, I don't think so.
Okay, just curious.
We missed that drought. Yeah. He's gonna say it.
I know. Well, we are, Rosemary.
Yeah.
Good.
Thank you.
So the regeneration the year after it rains. Is this, because the good growing season, these montane trees who aren't putting on exceptional amounts of growth are pouring their energy into cones and seeds?
Yeah, it would be nice to get somebody out there collecting that level of data too. It would tell us a lot, like masting and so on, is that happening during those years or afterwards, but we don't have that level of information. We just have the regeneration and the mortality and the tree growth. So anybody want to come join me?
Look at the especially it's flipping.
Yeah.
Is there any other studies you can sort of fudge and get that? That sounds really interesting.
You mean like model it out geographically?
Yeah, but other people got data relatively similar.
Well, there's a lot of research in Southern California, the Southern Sierra, and the type of data that we collect, it is spatially explicit, but there isn't a lot of the fine scale data feeds into the broader, context, but there isn't that level of data that we collected. There aren't that many studies giving that much depth. So I would say 54 sites across Northern California probably is enough to spatially explicitly, to model that, but it does infer something about these forest ecosystems. We could say that something about the forest ecosystems. I feel pretty comfortable about that generally speaking, but I wouldn't model it spatially as a geography. It's a good question. But we can't compare it to Southern California.
Well, let's give these lines.
Very much. And thanks for sticking through all those graphics.
And you can, if you want to chat more about graphs, you can always find our emails on the Cal Poly Humboldt website. We're pretty easy to find. You can shoot us an email. We do email. so sorry if I wasn't supposed to do anything. Okay, okay. I just.
Barbara, can I, listen, try out my machine?
And try what?